Okay, so we'll go ahead and finish up the digestive system. Am I remembering correct that we're right here? The liver? Okay, I think so too. All right, so we just finished the pancreas, right? We're talking about how the pancreas releases digestive juices, right, into the duodenum. And the pancreas has a couple different functions. One, there are a bunch of enzymes, right? Enzymes that digest absolutely everything under the sun. From nucleotides to proteins to lipids to carbs, everything. But the pancreatic secretions are also important because they have carbonates. And what do carbonates do? What do they neutralize? Acids. Now remember that the chyme coming from the stomach into the duodenum is very, very acidic. And we need to neutralize that acid right away before it goes on to the rest of the small intestine. So we utilize the pancreatic juices, the carbonates that are present there, to neutralize that acid. So now we'll talk a little bit about the liver. The liver is the largest glandular organ at over 3 pounds, and the liver is really versatile. We've talked about it quite a bit as it is, right? We talked about it when we talked about the blood. We talked about it in lab when we talked about hepatic portal circulation. The liver has a lot of different functions. We're going to focus on the digestive functions really here in this chapter, but let's just go through these really quick. So the liver is really important for different metabolic functions. And remember, metabolism is just like chemical reactions, right? Either building things or breaking things down. So what happens when all of our nutrients from the intestines are absorbed, remember hepatic portal circulation brings that nutrient-rich blood to the liver. And in the liver is where we process all of those nutrients. So we take things like carbohydrates, right, like sugars, glucose, and we store them as what? How do we store glucose? Glycogen, right? The liver links all of those glucose molecules together to make glycogen. It processes lipids, it processes amino acids in order to make proteins. And then it also can break them down as well. Remember, like when our blood sugar is low, what happens in the liver? Yes, beautiful, thank you. Glycogenolysis, right? It breaks down that glycogen in order to free up that glucose. Those are all chemical reactions. Okay, that's all metabolism. So we process a bunch of our nutrients in the liver. The liver also cleans out the blood, right? So it removes waste. Like remember when we talked about red blood cell recycling, right? And the bilirubin, remember? It gets broken down. And we utilize the liver in order to break down that and remove that bilirubin. We inactivate some drugs, so drug metabolism, your conversion of drugs either to an active form or an inactive form can happen in the liver. Phagocytosis and antigen presentation, again, we talked about that when we did lymphatic. Plasma proteins, remember, are made in the liver, and these are important for transporting things, for antibodies. They're important for the osmotic pressure when we look at capillary exchange. And then we also remove circulating hormones and toxins or whatever else. So essentially we're cleaning out the blood and then we're metabolizing nutrients. Now the liver also has a digestive function. Again, that's what we're going to focus on like right now for this lecture. The liver produces and secretes bile. And after the liver secretes bile, it gets stored and concentrated in the gallbladder. So when we look at the liver in the liver's digestive function, we often look at the liver and gallbladder together because they do work hand in hand. Okay, we'll see that the liver continuously throughout the day produces bile, but we don't always secrete bile into the duodenum. That bile goes into the gallbladder, we store it in the gallbladder, and then it gets secreted from the gallbladder into the duodenum. I'm out of breath, I don't know why. I thought I had a DVT the other day on the plane, so if I get a PE, somebody call 911 and tell them I'm having a PE. Seriously? My valves are messed up in my leg, so the blood pools. Oh, goodness. Okay, so here we see the process of producing and storing bile, and then secreting that bile into the duodenum. We'll see that we have a couple different hormones that come into play here. Secretin, which we mentioned in the first part of this lecture, stimulates the production of bile. And then we'll see cholecystokinin, which we have CCK right here, stimulates the release of the bile. Okay, and that release involves, it's listed here, but the contraction of the gallbladder and the relaxation of the sphincter here. So we'll mention both of those hormones as we go through this. So what organ did we say actually makes the bile? The liver, right? So the liver is constantly making bile. It says about 1 liter a day, but it's just throughout the day. It's constantly cleaning out the blood, and those waste products go into the bile, and it's constantly recycling or reutilizing bile salts. So all throughout the day, no matter what, it's making some bile. Sometimes you could stimulate it to make more, you can make less, but you're always making something. But when there's no food or no digestive components coming into the duodenum, we don't need to release bile into the duodenum. That would be a waste. So this sphincter down here, remember the sphincter of hepatopancreatic ampulla, or what did we call it? The sphincter of Oddi. This sphincter is contracted. So the liver makes bile, and it collects in, remember, the right and left hepatic duct, and then that unites to form the common hepatic duct, but that bile cannot actually be released down into the duodenum. So it just follows the path of least resistance, and it goes up into the gallbladder. So the liver produces bile, and then it's stored into the gallbladder. As it remains in the gallbladder, it becomes concentrated. The reason for that is that we just reabsorb water out of the gallbladder. Okay, the gallbladder is kind of similar to our urinary bladder in that it's the sac that expands greatly. Okay, so as your urinary bladder fills up with urine, it expands. As your gallbladder fills up with bile, it expands. Okay, and it can just store more and more and more and more bile until that bile is needed. And again, we reabsorb water so that the bile becomes really concentrated, and that increases its strength or its usefulness once it's secreted. So when we have chyme go into the duodenum, remember, so here's our small intestine, the first part of the duodenum, and remember that that's hooked up to the stomach, right? We've got the pyloric sphincter right here. When chyme comes into the duodenum releases cholecystokinin. Okay, that's our hormone right here that we said is important for the actual release of bile into the duodenum. What that cholecystokinin does is it comes up here and causes a contraction of the gallbladder. Okay, so the gallbladder actually contracts to squeeze out the bile, and it causes the relaxation of our sphincter of Oddi. So now the bile can actually be released down into the duodenum. Okay, so the stimulus for the release of bile is chyme making its way into the duodenum. Okay, that tells the gallbladder, okay, I've got something here. I need you to release your bile so that we can work with this chyme. We can digest it further. So the bile itself has a couple different functions. One, bile has wastes that need to be excreted from the body. Remember, we said the liver cleans the blood, right? The liver removes drugs, it removes toxins, phagocytizes old cells, bilirubin. We've got all sorts of wastes that are removed by the liver. And we need to get those wastes out of our body somehow, right? The liver doesn't directly connect outside of the body. So what it does is it puts those wastes in this bile, and then they eventually make their way into the intestines. And if you follow the intestines all the way down, and then around the large intestine, eventually you make your way out of the body. So that's how a lot of wastes get out of the body in feces, via the bile. Bile also helps in digestion though. Bile has something called bile salts, and bile salts help to break up these huge lipid droplets into smaller little pieces of lipids that we can digest easier. And we'll see that on this next slide. So bile is made by the what? Liver, and released by the gallbladder. When we look at bile, we said that bile has wastes that we need to get rid of, and bile also has bile salts. And bile salts are important in digesting lipids. remember that lipids are not water soluble, right? What do we call them? Hydrophobic, right? They do not like water. So when we look in the, like in the stomach, when we start to squish around and break up and mechanically kind of like just mix up all of our dietary components with all of the water that's in there, we see that the lipids tend to congregate together. They don't want to disperse into that water because they're hydrophobic. So the lipids all group together and they form these nice large droplets. So we have these big drops of lipids. Now, the problem with this is that when this chyme moves into the duodenum, remember we've got a bunch of lipases that come from the pancreas that break up those lipids. These pancreatic lipases are not lipid soluble, so they will not enter into the center of the drop. Remember we talked about this when we talked about the surface area and how important it is to get a greater surface area of the food molecules? So if I have a huge lipid drop like this, right, a whole cluster of lipids, the pancreatic lipase can only work at the outer edges. All of this in the center is protected and won't be digested. Now the bile salts come in and they interact with all of these lipids so that the lipids don't cluster together anymore. In essence, they give the lipids solubility in water. So now the lipids can disperse throughout the water and we have tiny little droplets. And now we have a much greater surface area for the enzymes to work. So we don't have all of this area that's not going to get digested anymore, and the lipids can get broken down a lot easier. Breaking these lipids apart into these smaller little droplets, that's called emulsification. Okay. Once we break it into these droplets, we coat them with bile salts. And again, like I just said, that gives them solubility, so that keeps them in solution so that they don't just rejoin up again. So almost all digestion and absorption occurs in the small intestine. Remember, when we looked at the stomach, we started to break down some nutrients, but we weren't absorbing anything, right? No absorption was occurring in the stomach. Once we get to the small intestine, specifically the jejunum, we see that we have a ton of digestion and a ton of absorption occurring. Intestinal absorption is just when we see nutrients or vitamins or minerals crossing from the GI tract into the cells that line the GI tract, right? The columnar cells that line the GI tract. And then from there, they enter into the interstitial fluid surrounding the GI tract, and then eventually into either the blood or the lymph. Okay, we want to absorb these nutrients from the GI tract into the body fluids. So we have a tube, they're in the middle of the tube. First they cross into the cell that lines the tube, then they cross into the interstitial fluid, then they can enter circulation. Okay, either a blood vessel or a lymphatic vessel, depending on what kind of nutrient it is. So when we look at absorption, we see that different cells are absorbed, different nutrients are absorbed in different ways, right? Because they have different characteristics, different solubilities, different sizes. We see that monosaccharides and amino acids enter via facilitated diffusion and co-transport. Remember that co-transport is when you move two things in the same direction at the same time. We see that most glucose and most amino acids are transported by sodium, glucose, or sodium, and whatever amino acid we're looking at, transporters. So these are transporters, proteins on the membrane that grab both sodium and glucose, or both sodium and an amino acid from the lumen and bring it into the absorptive cell. And that's how we move most glucose and amino acids. Lipids, remember, have the ability to go directly through plasma membranes, right? When we looked in the small intestine, we saw that we broke them up into tiny little droplets, and then we let our lipases break the lipids up into fatty acids. And these fatty acids are small, and they're lipid soluble. So they have the ability to diffuse directly into the absorptive cells, right? We don't have to use any facilitated transport. We don't need to use any proteins or transporters or ATP or anything whatsoever, right? They just go directly through the plasma membranes into the absorptive cells. Once they're in the absorptive cells, we repackage these triglycerides. Okay, so we make triglycerides and repackage the lipids. So we'll take the fatty acids, we convert them to triglycerides, and we essentially just put a bunch of fatty stuff all together. And we call that unit a kilomicron. So kilomicron, again, is just fatty stuff. So we have triglycerides, we have some lipid derivative hormones, we have lipid soluble vitamins, any type of fatty acids, cholesterol, any fatty component will get packaged into this big kilomicron. And then that kilomicron undergoes exocytosis out of the cell into the interstitial fluid. And then from the interstitial fluid, it'll enter into the lacteal. So if you remember when we talked about the small intestine, remember the small intestine, the mucosa has these villi like this? And in the center of the villi, remember we have a lacteal. And we said that a lacteal is what? Good, a lymphatic capillary. What's a major difference between lymphatic capillaries and blood capillaries? Something that might matter for the absorption of these really big kilomicrons.
The permeability.
Permeability. Remember that when we look at the lymphatic capillaries, that their cells overlap like this, right? And they provide these nice large openings so that big cells and big things can enter into lymphatic circulation. Because remember, we want like bacteria and stuff like that to be able to enter easily. So that also means that large things like kilomicrons, these big balls of fat stuff essentially, can enter into the lacteal. So that's why we have those lymphatic capillaries in the center of the villi that stick out of the small intestine for fat absorption. When we look at vitamins, most vitamins are going to enter cells just by diffusion. They're relatively small. They're enter by diffusion. However, the fat-soluble vitamins will get packaged with the fatty acids in the kilomicrons. Right, which we said right up here. All right, where is it? Lipid-soluble vitamins will get packaged in with the kilomicrons. So the fat-soluble vitamins, you guys should know this, include A, D, E, and K. ADEC. Vitamin A, vitamin D, vitamin E, vitamin K. This is important because these are the ones that can actually build up in your body. They're like fats, so they'll get stored in your fats, and they'll build up in your body if you take too many of them. The water-soluble vitamins, like vitamin C, you could take 10,000 times the daily requirement and you would just pee it out because it's soluble in water. But the fat-soluble ones will build up in your body. Vitamin B12, remember, was special. Vitamin B12 needs to bind to intrinsic factor. What organ did that come from? Intrinsic factor. The stomach. Intrinsic factor comes from the stomach. And vitamin B12 has to bind to that intrinsic factor before active transport can transport it into the cells and then into the body. Water just follows the stuff, as always. Right? Water just follows the osmosis. So as we're absorbing all of these vitamins and nutrients and all of this stuff, the water just follows along and gets absorbed. Okay, so it's just passive absorption. Brings us to the large intestine. When we look at the large intestine, the key kind of functions or purposes of the large intestine, one are the reabsorption of water. Remember, we have put a ton of water into the GI tract. From the 1.5 liters of saliva to liters in the stomach, liters in from the pancreas, from the small intestine. we have liters of fluid going into the intestines. And think about it, when you poop, do you poop out liters of water every time? No, not unless you're really sick, right? You shouldn't be doing that. So that water has to come out of the GI tract and back into the body somehow. That happens in the large intestine. Okay, typically in the very first parts of the large intestine, we reabsorb the majority of the water that we put in. We also compact all of the intestinal contents into feces. Again, you don't poop out this liquidy, milkshakey, watery stuff. You poop out a nice formed stool if you're healthy. And that's because the contents get packed into this nice formed feces as we reabsorb the water out of the large intestine. We also absorb some vitamins. There are some vitamins, vitamin B5, vitamin K, biotin, that are actually produced in the large intestine. We have healthy bacteria that live in our large intestine that are supposed to be there, that break down the last little kind of components of the food that are left. And as they do that, they produce vitamins. Since they're just now being produced, we have to absorb them now. Right? We can't absorb them in the small intestine because they didn't exist yet. We produce them in the large intestine, so we have to absorb them in the large intestine. This is really all of the absorption that's going to be happening in the large intestine. Most absorption of nutrients has already happened in the small intestine, okay, almost all of it. We have water and then these last few vitamins that we're going to reabsorb from the large intestine. Also, we store fecal material prior to defecation. And again, this is very nice for convenience, right? We don't constantly leak out waste products, but we store them in the large intestine until we're ready to release them mass movement at a time. So when we look at the lining of the large intestine, we see that it differs from the small intestine in quite a few ways. One, it lacks villi. Where are villi present? Where? Small intestine, right? Villi, those little finger-like extensions like that, they're only present in the small intestine. They are not present in the large intestine. Also, when we look at the large intestine, we see these really big, deep glands that produce secretions that are just full of mucus cells. Okay, so when we look at these glands that are present in the large intestine, we do not see any enzymes being produced. We have no enzymatic breakdown occurring in the large intestine. Okay, all of the enzymatic breakdown occurs from the saliva to the stomach to the small intestine. No enzymes get released into the large intestine. So any of the breakdown that's going to happen in the large intestine is going to be due to bacteria, which we'll talk about in a second. The mucus glands that are present in the large intestine are important to provide lubrication for the fecal material. Remember, all this time we've had this really kind of watery stuff that's been swishing through the stomach and the small intestine, and now we reabsorb all that water. So you have this dry stool, and you need some way to make it slide nicely through the intestines. So a nice thick mucus coating is important for that, to provide that lubrication. The longitudinal layer of the muscularis externa. Remember the muscularis externa, that muscular layer? Remember like the mucosa, submucosa, muscularis externa, then serosa. The muscularis externa typically has two layers, right? Circular muscle that goes around like this, and then longitudinal muscle that goes down along the length of the tube. When we look at the large intestine, that longitudinal layer of muscle is reduced to two thin little bands. And remember the Taenia coli that we saw when we looked at the large intestine in lab? Those two little like white lines that run on either side of the large intestine? That's what's left of the longitudinal layer of muscle, just that thin Taenia coli. And we'll see that that's important to kind of cinch or pull the large intestine up to create those little pouches or haustra that we saw in lab. Again, when we look at the large intestine, less than 10% of nutrient absorption is occurring here. Because where did most nutrient absorption occur? In the what? In the small intestine. Did any of it occur in the stomach? No. No. Okay, so most nutrients are absorbed in the small intestine. Less than 10% of nutrient absorption is going to happen in the large intestine. However, we do see a ton of reabsorption of water. Again, liters of water are reabsorbed in the first part of the large intestine. We also see the reabsorption of bile salts in the cecum, the little first part of the large intestine. we reabsorb those bile salts. Remember we said that the liver just recycles them to make new bile. So we'll reabsorb them in the cecum, they'll get transported to the liver makes new bile with them, and we break down new lipids. We have the absorption of the vitamins that are produced in the large intestine. Remember, we do not have enzymatic breakdown in the large intestine, right? No enzymes are breaking anything down, but we do have healthy bacteria present. Okay, so there are bacteria that are part of your biome, right? They're part of your little body environment. The bacteria are healthy, they're supposed to be there. You need them to function appropriately. So these bacteria that are present in the large intestine will break down the last little kind of components of the food, and in the process, they produce vitamin K, B5, and biotin. Okay, we have to absorb them from the large intestine, otherwise they would just leave in the feces. So we produce and absorb them in the large intestine. This is what you're replacing when you take probiotics. Like, you know, when you take antibiotics, and sometimes it causes diarrhea, that's because the antibiotics also kill your healthy bacteria in your GI tract. And then that causes the antibiotic-associated diarrhea. So that's why they say to take probiotics with it, or yogurt that have active cultures. You're trying to replace those healthy bacteria that the antibiotics would kill. We see the same thing like in the vagina. You can kill healthy bacteria that are supposed to be there, and then yeast overgrows because it doesn't have competition with the bacteria. Okay, so some bacteria are healthy in your body. This is kind of an odd one, the absorption of some wastes, and that sounds really silly, right? Why would you absorb wastes back into the body instead of just letting them go out in the feces? But remember, like when we talked about bilirubin breakdown, how the bilirubin is taken out by the liver and put into the intestines, and then some of it is converted to urobilinogens. Remember, I didn't make you memorize that part, but it's sterco and urobilinogens. The sterco stay in the feces, the uro get reabsorbed and then excreted by the kidneys. That's not a huge important function. We have multiple different movements that we see relating to the large intestine. Last lecture, we mentioned the gastroileal and gastroenteric reflexes, and these are just smooth muscle contractions that move materials into the cecum when you eat. So essentially, you eat food, the stomach expands, that food is in the stomach, it's going to get pushed into the duodenum, and that tells the body, I have a huge amount of food coming and I need to make room for it. So we move everything from the stomach along the small intestine, and from the small intestine into the large intestine. So we ultimately get materials into the cecum when we're eating a meal. This is why in the healthiest functioning GI tract, when you eat a meal, you should have to poop soon. Because you're eating a meal, it pushes everything forward, everything moves down the line, and that should stimulate the need to release something from the GI tract to make more room. So then once we get into the cecum, we have movement from the, so remember like small intestine, and that goes into the cecum. And then we have ascending colon, transverse colon, descending colon, sigmoid colon, and then rectum. Gosh, that's beautiful. That's really good. Love it. Okay, so movement from the cecum up the ascending colon to the transverse colon is very, very slow. Okay, it's peristalsis, which remember is like a wave-like contraction that pushes things forward, but it is weak and it is slow. It takes hours for things to move from the cecum up through to the transverse colon. The reason for that is that that is where most water absorption is occurring. So we need a lot of time to take the water back out of the GI tract and reabsorb it back into the body. Then we have peristalsis that occurs around the whole rest of the colon that's a lot stronger and faster. Okay, we'll talk about that on the next slide, but that has to do with when you actually get the urge to defecate, when we have these big, massive movements. But this initial peristalsis is very, very slow for water reabsorption. We also have another kind of movement occur in the large intestine called segmentation or haustral churning. What's a haustra? Or what are the haustra? The rings on the large. These little segments, right? So like these little, how it's organized into these little groups like this. Postural churning or segmentation is when the contents or materials just get swished back and forth like this. So when stuff just goes from one hostra to the next, one hostra to the next like this. So you're not actually moving anything forward like you are with peristalsis, you're just mixing, okay, back and forth between the two neighboring hostra. So I said that movement from the cecum up to the transverse colon was really slow. Why is it so slow? Water absorption. Movement through the rest of the GI tract, so from the transverse colon and then down through the rest of our colon, occurs because of big, strong, rapid, powerful peristaltic movements. We call these mass movements. And these mass movements, again, are stimulated when we have distension of the stomach and duodenum. So we just said, what happens when you eat a big meal? You should get the urge to go poop. Okay, the reason for that is you eat a big meal, the stomach distends, stretches, and it sends signals along the nerve plexus of your intestines, so the nerves go all over your intestines and go to the large intestine, and it stimulates this strong, powerful peristalsis that pushes everything forward in the GI tract. Because you just ate a meal, you need room, right? Everything has to move forward if you're gonna fit more stuff into your intestines. Once the contents get pushed into the rectum, which remember the rectum is like these last 5 inches or so of the GI tract, that's what triggers that defecation reflex. Okay, so what I mean by defecation reflex is essentially the urge to defecate, but we'll talk about it more right here. Remember when we look at the GI tract, we see that the very end of the rectum here is guarded by 2 anal sphincters. We have an internal anal sphincter, which is smooth muscle. You do not consciously control it. And then we have an external anal sphincter, which is skeletal muscle that you can consciously control. When the feces gets pushed into the rectum, that triggers this, this reflex, this defecation reflex, that relaxes the internal sphincter. Remember, you don't control that one. It relaxes it and it tells you that you need to go. It gives you that urge, oh, I need to go to the bathroom, I need to poop. And it also allows you to constrict the external anal sphincter. You will not release feces until both sphincters are relaxed. And you consciously control the external anal sphincter because that's skeletal muscle. Okay, so that's why you can control the actual defecation because you consciously will relax that external anal sphincter. I cannot.